perhaps you would also like to say a few words about our idea.
Yeah, thank you, Ralf. We've been working on this medical omics task and setting up a group that will be interested in this for a couple of years. And all these years there have been complications, but I hopefully will overcome them in the next years. The most important thing is that we believe that medical omics will fuel the precision medicine that will come in the next decade or so. And therefore, I believe it is important that we put this medicines on a very good foundation so that we don't get precision medicine that is not informed in the best way possible, but with the way it would be informed with bioinformatics now, which is rather foggy. Great, so much for that. And with that, I want to invite Yusuf Tuta, our first speaker of the day. He asked him where he was currently, but I didn't have time to check that. So he is in Istanbul, and yes, he's located in Turkey. He has been working on cancer and different topics in the medical area for a long time. And we are very, very happy that he brings to this workshop a different perspective, not just a computational perspective, but also a practical perspective. And that is what I think we need in order to be successful at the end. So thank you very much, Yusuf.
Okay, so I would like to thank, should I start my presentation now or just...
You can introduce yourself, you can start your presentation directly as you like.
Okay, so I wish to be there alive, but as you said, we had problems with COVID. Well, it's fine, and actually, one part of our problem in cancer research is there are huge amount of data. So we don't know how to deal with all those, but bioinformatics is especially useful for us. So as I will present in my research data, we are having problems in relating gene expression data and metabolomics data. So I'm hoping that at the end of my presentation, I can get your help, and maybe that will make my research much easier. So I want to thank the organization committee for inviting me over to this lovely meeting. It's a really nice group, and I'm hoping that we can make some collaborations in the near future. So today here in Istanbul, it's a rainy day, but I was supposed to be at Iran. One of my friends invited me to another conference. So because this conference is canceled, I thought, I'll be having a hard time because of these chaotic events in Iran. But they canceled the Congress, and right now I'm in Istanbul, and I'm hoping that the internet connection is fine. And whenever you want, I can start my presentation.
Please go ahead. Thank you.
Okay, I hope I can. Let me see. This is, well, I'm not very familiar with this system, but I'm hoping that...
Share button or arrow that points up. Yeah, perfect.
Yeah, you see the screen? You see the slide, sir?
Yes, we do.
Okay, so I'm working at University of Health Sciences. It's a university that is a bit different than the rest of the universities in Turkey because the university is facilitated with 58 hospitals. The association provides some benefits, both it's a mutual benefit, but the university has a campus outside the Turkey, and the university is very complex, but especially for studying illness or medical sciences, you may easily get samples, patient. In this way, it's easy to work in the campus, but it's a huge and a complex structure. So I work at Faculty of Pharmacy, and I'm a biochemist, and I also teach at a molecular oncology program. And I know Jens. He's a super scientist. He's a super eminent scientist, but he chose to leave. It's a loss for Turkey, and he's a great bioinformatics scientist. So I'm thankful to him as well. And today my topic is targeting HSP90 and co-chaperones for breast cancer drug design. And I will start from protein folding. The primary structure of the proteins is easy to understand because it's basically formed from different residues. But the problem is how these primary structure folded to the tertiary structure. Although we know the solution, which is water, there are other factors that will influence the folding of the protein. So in a test tube, it's much easier because there's nothing that will restrict the folding. But in the cellular environment, this folding is not that easy because there are other key essential factors that will influence the folding. So I'm not going to talk about the potential of folding, but we know that folding of the proteins in the cellular environment is not easy. Well, if we are working on the lower concentration at lower temperature with a small peptide, this is easy because there are less things that perturb the protein folding. But generally, all the proteins are larger than these small ones, and they need special assistance. And this assistant is important, especially at elevated temperature. So there are cells develop proteins like heat shock proteins or called chaperones that will help the substrate protein folding. So this makes function easier because there's a direct relationship between structure and function. So if the protein is not properly folded to its native state, then
Yusuf, are you showing slides? Because we only see the first slide.
Oh, I'm on the third slide. I'm sorry. This is
perhaps you have two screens and you're sharing the one that shows. Yeah. So this we see.
You see the you see the slide right now. I'm sorry.
Right. We now see your PowerPoint with the slide selection and the third slide selected.
Okay, so maybe I should use these what I did is I used the full screen, so maybe I should go this way. If you see the third slide, then it's fine. I'll go with that one.
Yeah, okay, go with that one.
Okay, sorry for the inconvenience. Well, this is important because we know that the function of the cell depends on the protein. So proteins functions like cell division, apoptosis, all of these, or signal transduction, all, or immune, like we call it, for the immune system or defense system, we need functional proteins. And to get functional proteins, those proteins must be in their native state. So since the native state is important, the heat shock proteins or chaperones are essential structure for the cell. So there are around 100 heat shock proteins or chaperones that help either protein folding or other functional biochemical processes. Molecular chaperones are required in protein folding for sure, but they are also useful in translocation, assembly, and degradation because there are different compartments in the cell, like transferring a protein or extracellular protein. Heat shock proteins comes into play. Same thing for transforming from one compartment to other. And also, heat shock protein play key roles in immune system, not only helping folding, but they also induce T cell activation. And you can see from the slide, these actually are essential roles. And actually, this inducing T cell response is one of the projects in our hand right now. And also, heat shock proteins play key roles in apoptosis and DNA damage. Because once the protein is not useful, then substrate proteins send it for degradation. Because as you know, these the biochemical system needs nitrogen, so unfunctional proteins will accumulate, so that will be problem. So what molecular chaperones do? They well, in the presence of stress, the situation is different, but generally 50% of the proteins go through the simultaneous folding. But 20% of the proteins needs folding. And the rest of the 20% goes to degradation. And actually that process is also coordinated by heat shock protein or chaperones. So heat shock proteins are actually a large family. They have diverse function. They have several families and named with their molecular weight: heat shock protein 40, heat shock protein 70, 160, and small heat shock proteins, as well as heat shock protein 90. And these proteins generally work with coordinating and coordination fashion. So heat shock protein 70 may complex with heat shock protein 90. Actually, the combination of different families forms distinct functions. Yet we don't know much details of those, but we know that there are at least 11 HSP70, 5 HSP90, 6 HSP40. So we have not only folding at cellular environment for different organelles, there are different heat shock protein isoforms. For example, heat shock protein 70 isoform in cytosol is distinct than the heat shock protein 70 in the mitochondria, and they also have 4 induced state and constitutive state. So all these families somehow coordinates and function so that the proteins in the cellular environment are in their native state. So as I said, there are different states of heat shock protein family, and actually each function is distinct. Like HSP 100, well, by the way, the proteins are basically, most of them are ATP dependent, and HSP 100 is disaggregate and unfold the aggregated proteins. And HSP 90 has a huge cavity, is a dimer protein, and provides hydrophobic environment. If you imagine the crowding of the cellular environment, like HSP 90 is a fitting room, it's a unique place. So substrate proteins enter to this fitting room because it's not affected from the environment, so that protein may fold easily, and that cavity or the fitting room is basically formed from hydrophobic residues. So these the environment provides hydrophobic forces to fold substrate proteins. And actually, HSP 70 provides pre-folding process. HSP 70 process 7 residues of the substrate protein and then submit it to HSP 90. So these two works in combination, and this combination actually makes the protein folding easier. And of course, there are cytosolic heat shock proteins, and well, these are on the textbook like GRO-EL and GRO-ES, and these are barrel-like structures, and HSP 10 or the GRO-ES is the cap that covers the top of the fitting room. That's my analogy, because those are places that substrate proteins fold to their native structure. And because of these folding processes, there are several studies done, especially in neurodegenerative diseases, because we know that heat shock proteins play key roles in, for example, frying infected cells. And actually, in my second postdoc, I worked with these accumulated proteins, and they form long fibers. And heat shock proteins are essential to remove those highly complex protein accumulation. So generally, scientists use model system like yeast or bacteria, and as you can see from the slide, heat shock protein 100 plays key role in dissolving these large aggregates, and the complex get the help of HSP 70 and HSP 40. So they help unfolding of the substrate. So those are the models. So why we try to use heat shock proteins as the target of cancer studies. We know that heat shock proteins also play key roles in cell survival. So heat shock proteins inhibit apoptosis. So what happens is, if the idea is if the heat shock proteins are inhibited, then cancer cells can be driven to apoptosis, and this will help anti-cancer studies for drug design. And actually, there are several groups that work on those drug designs, but let me tell you about the drug design a bit. So, like 10 years ago, people used to screen 10,000 to 20,000 candidate drugs, and then they do some in silico techniques to understand these absorption, distribution, metabolism, expression, and toxicity studies. And then they go to clinical trials, and then the FDA approval, and then at the end of 10, 20 years, you get one drug to the market. But right now, this is much easier because we luckily have scientists on bioinformatics, and we can use big data, and we can illustrate key information targets, and from here we can easily design new compounds. So actually, this slide is a bit old, and rather than for 10 years right now, the time frame is maybe shortened to a couple of years. And with the help of bioinformatics, we can any research group can join to these cycle from anywhere. But generally, when we screen the genes, we have a profiling, and then we screen these profiling in the presence and absence of the candidate compounds. And if the compounds are displayed anti-cancer activity, then we use animal experiments. And then if that animal experiments are they provide a good results, then that compound starts the clinical trials, and at the end of these clinical trials, a set of drug is available for the market. But we know that the side effects or other off-target effects are important. So at this point, you can use the clinical drug and search for other targets. So this is also beneficial because you may use the same drug for other purposes. So drug repurposing is actually another field. So you can use this cycle and enter from any part to start. So back to the heat shock proteins. So we decided to understand how heat shock proteins behaves. But the problem is because heat shock proteins have different isomers. So for each isomer and each condition, one needs to test whether these heat shock proteins display intrinsic or extrinsic apoptosis. So for that, especially at NIH, Lannecker start using inhibition of heat shock protein 90. So why heat shock protein 90 is important? Because this protein is actually up to 2% of all our cellular proteins. And heat shock protein 70 is overexpressed in tumor tissues, and this may go up to 10-fold. So that's why it's a fine target. And actually, heat shock protein has a lot of substrate proteins, and I'm not going to go through all of these, but you can see that the substrates actually, it's around 400 to 500 substrates. And though some of those are important, and like the CDKs or cyclin Ds, that they are involved in the cell cycle in here, you can see the signaling in the cell cycle, and the ones that is bold is actually the target of HSP90. So, for example, AKT is the substrate of HSP90 because it interacts with CDK4. Or the receptors of the breast cancer is also the target of HSP90. So the idea is, if you inhibit HSP90, the folding pattern of these essential substrate proteins will not reach to their native state, so they will not be functional. So this will drive the cells to apoptosis. So since the substrates needs to be folded to their three-dimensional shape, HSP90 is essential. So HSP90 binds to these substrate proteins and fold to their native structure. But if we inhibit HSP90 with the inhibitors, since the substrate protein will not fold to its native structures, so these will send it to proteasome for degradation. So that's our purpose. And actually, heat shock protein 90 is a dimer, and the cycle works with ATP, and ATP hydrolysis open the fitting room, that cavity, and the substrate proteins penetrates into this cavity, and then it helps folding of the protein. So that's why some clinical drugs are used, especially coumarin derivatives, to inhibit HSP90 function. And Lanneckers actually find geldenomycin for HSP90 inhibition. And HSP90 is inhibited with geldenomycin, but the problem is that drug has some solubility problems along with the hepatotoxic effects. So we decided to design a NIV inhibitor, and we were hoping that it will be less toxic. So this is the dimer of HSP90, and the green one is the inhibitor, and actually you can see the two different domain, and the inhibitor binds to the hinge region of HSP90. And actually we did we use some in silico studies to understand if that really binds to these hinge region. And we did several set of experiments to understand, not by using modeling, but we did cell culture, microarray, and we did some mutational work and some biochemical assays along with the binding assay. So it's difficult to visualize that in the previous slide, but in here you can see that actually HSP90 is a dimer that has N-terminal domain, middle domain, and the C-terminal domain. And that hinge region that's shown with the red arrow is the target that our molecule actually binds, and it's a coumarin derivative as well. And in the absence of our inhibitor, the dimer forms a nice fitting room. You can see this fitting room and the substrate protein inside the fitting room, and that will help the folding pattern of the substrate protein. But if we add our inhibitor, then the dimer will not form and there will be no fitting room. So the substrate proteins may not fold to the native states. So that is the idea. But so what we did here is that the problem here is everything were found in the test tube. So we have HSP90 inhibition, and we used these in the animal studies, but didn't work, and we didn't understand at all. And because the inhibitor works very well, and mutation works shows that inhibitor binds, and but the problem is we did some expression studies, and we found out that HSP70 actually complements the function of HSP90. If you inhibit HSP90, this induces more HSP70. And this HSP70, even though folds substrate protein less efficient than HSP90, this increase in HSP70 actually complements the absence of HSP90. So this shows the coordination of HSP70 and HSP90. So HSP90 and HSP70 coordinates and cooperates together. So even though you inhibit HSP90, increase in HSP70 will complement the function. So the structure of HSP70 is actually a bit different than HSP90, but basically there's an ATP hydrolysis domain, and then there's a substrate binding domain, and as you can see that small yellow substrate penetrates into the substrate binding domain. So that also provides a fitting room. This is way smaller than HSP90, but still coordination of HSP70 and HSP40 do some good job and actually complements HSP90 function. So what we did is we compared the structures of HSP90 and HSP70 so that we can design a compound that will inhibit both HSP90 and HSP70. So the reason we didn't use ATPase inhibitor because of these off-target effect, and we screened lots of inhibitor, and we designed a coumarin derivative, and that actually will fit in both to HSP90 and HSP70 inhibition. Well, also, luckily, HSP70 coordinates with several other co-chaperones, and these co-chaperones are actually forms different functions. So our inhibitor actually is not only bind to the substrate binding domain, but also binds to the interface of these co-chaperones. So we basically inhibit the protein by using two different processes. So in our projects, what we do is we use in silico techniques, but those are limited to docking studies and simple simple mechanistics. We are not doing dynamics yet, and actually the rather than using a whole set, we use only a small set of genes, around 80 genes. So we start with cell cytotoxicity and support these with in silico experiments, and then array experiments, and then we do some cellular work like programmed cell death, senescence, or cell cycle experiments using flow cytometer. And then we also support these by binding and stability experiments with the structural biology. And then we are using animal experiments to understand the effect because cellular lines are not effective, especially to understand the metabolite and especially the sugar metabolism. So this will help us understanding the effect that animal models. So basically, we use two groups. We grow the tumor, and then we excise the tumor in one group, and we use both groups in testing our compounds and to understand the preventing and the treatment effect of the compound. So we have right now a patented drug that really works efficiently, and we also did some comparison studies with the clinical drugs, and of course we used the healthy cells, and the compound did not show toxic effects. I skipped those. Also, we have we tested the compounds at resistant cell lines, and some of them are really effective in these resistant cell lines. Also, we use imaging system, and our compounds are actually really effective on cells at lower concentrations, and the toxicity is well at the lower level. So we are lucky, and this actually shows an in silico studies that compares our compound with the commercial inhibitor, and this inhibitor actually our inhibitor binds the substrate binding cavity, but these commercial one not. And also our inhibitor binds to the interface of the co-chaperones. So this is not only inhibits the HSP70, it also inhibits the co-chaperones interaction with HSP70. So in this way, the compound is useful, and also the compounds affect both HSP70 and HSP90. This is the in silico binding studies of my collaborator. So this dual inhibition is actually provides a good yield for anti-cancer activity. And also we try to understand if the compound binds to HSP70, and we compare this with this commercial drug, and this commercial drug actually does not bind in the presence of ATP, but our compound binds in the presence or in the absence of ATP. And actually, our compound has an amine group that we can add chromophore, and we can trace if the compound really finds a target. So that's why these two properties nicely designed for anti-cancer studies. And of course, our compound does not change the secondary structure composition as well as the stability of the proteins that I'm not going to go into details. So the compound works pretty good as anti-cancer drug candidate. So we did several set of experiments, and we have around 250 compounds, and most of them is really effective at different cancer types. And but we also target other parameters because we want to increase the efficiency of our drug candidates, because we know that cellular organelles coordinates, and we did some microarray experiments, and we find out that pseudogenes, non-coding genes, as well as some of those isoforms and two microRNAs are important for cell survival in breast cancers. And in here you can see the start of the apoptosis, and cellular cytosolic HSP isoforms interacts with mitochondrial HSP isoforms. So as we can see in the previous slide in here, these interactions are important. So what we did is actually we induced one of these microRNA, and we observed that this induction drives the MC7 cells to apoptosis, and suppression of some of those pseudogenes are also effective. So using just single inhibition of HSP90 and 70 is not enough. And we also did some array studies to understand if, well, we do primary work in cell culture, and if the cell culture is effective, then we use a small set of genes, and we do some gene expression analysis, and at the end, we saw that these expression pattern, and we use gene expression analysis, and we can have an idea of the cell cycle or program cell death, or if the compound increases the immunity of the cell, or we can get several different clues about the gene expression. And actually, we use these gene expression, and we saw that actually that inhibitor use JAK-STAT pathway to inhibit. So, and actually, there are other ways that we can display how the compounds are worked, in which process in cell biology is enhanced, whether the cell cycle, cell growth, or apoptosis, or if the cells are driven to oxidative phosphorylation, we can easily determine by using these array studies, as well as the supporting evidence like the apoptosis, as you can see in here. So, and of course, we do some cell cycle analysis to understand which stage of the cell is affected with our inhibitor, and we use clinical drugs to compare with our compound. And of course, we use synergetic or additive effect to understand if the compounds can be used with the clinical drugs. And actually, in this case, this compound displayed an additive effect. So at the end of our experiment studies, we saw that we have to deal with the non-coding RNAs, including pseudogenes and the microRNAs. We have to understand the interaction of cellular organelles, and also the energy metabolism, because like in the triple negative breast cancer immune subtype, not only we see Warburg effect, we also see oxidative phosphorylation. So that's a bit hectic. And also we deal cell death, but currently we are working on cancer stem cells along with these resistant cell lines. So with these, we use different subtypes with different resistant forms, and we use aryl hydrogenol compounds. And if you can recall, the model system is HSP90, CDC37, and CDK4. Actually, CDK4 is essential phosphate, and CDC37 helps coordination of that compound with HSP90. So our compounds are actually inhibiting this complex, but still we have problems. So what we did is we, I hope I still have a couple minutes, right?
Yes, you have.
Sorry. And what we did is we also need to deal with the cancer cell heterogeneity. So we know that a subset of the cells are basically cancer stem cells, and we know that these affects our results. So what we use is we use 4 genes to screen if these stem cells has any effect. And we know that these stem cells has effect, but our compound, one of our compounds, basically inhibits the effect of these stem cells, and making a combination of compounds and using for specific subtypes of breast cancer actually totally inhibits the proliferation. So we are right now on the animal experiments, and the compounds displayed a pretty good activity, and one of them is patented, and 2 of them is waiting for the patent. So but at the end, we have a good candidates for breast cancer for different subtypes. And I would like to thank my collaborators and my PhD students, as well as my grant holders. And I also thank you guys for inviting me over to this lovely meeting. And I appreciate if you have any questions, and thank you for your attention.
Thank you very much, Yusuf. I do have a question immediately.
Yeah, please.
And you showed us some slide with cell viability. So I was just thinking about the off-target effects of this best drug candidate that you have that inhibits both HSP70 and HSP90.
Yeah.
So what are those off-target effects that were predicted and the ones that you see in the animal model?
Oh, the off targets. Well, we design. Well, for our case, we didn't see any off target effect. But when we design ATPase inhibitors that I didn't mention, because most of the substrate proteins has ATPase domain, so that the compound binds to those ATPase proteins. So that's why you need to use a more concentrated compound. So there are at least 15 ATPase proteins that binds to the compounds that if you design for ATPase inhibition. So that's why we didn't choose to use ATPase inhibitor, but we choose substrate binding domain inhibitor it basically binds to the cavity of substrate protein domain, and it inactivates HSP70. And in the case of HSP90, it binds to the hinge region, so HSP90 cannot form dimer. So we did not see any off-target effect, but we see other additional effects like the resistance or cancer stem cells or the pseudogenes and the microRNA. So we have to deal with all those to get an optimum drug. So it's a mix. I hope that answered your question. Sorry.
Yeah, I'm not 100% sure. If I were to shut off HSP70, HSP90, then I would significantly interfere with protein folding.
Yeah.
And that should lead to some effect in the mouse cells.
Actually, what we do is we directly inject the inhibitors to the breast. We did not give the drug orally or from the tail.
Okay, so you have a very targeted approach, and then
Yeah.
the off-target effect that you would see in healthy cells would not be so important. I see.
Yeah, that helped. Actually, our friends they are working on drug carrier systems. They try to design, but injecting directly to the tumor tissues is really help. And maybe that's the reason we don't see the off-target effect. But it's real. I mean, the anti-cancer activity is especially at lower concentration of these compounds are pretty good.
Thank you. Other questions? Oh, wait, I will turn you on. Nein, ist noch nicht. Do you hear him? Du musst lauter sprechen, damit er hier auch hört. So the question is, did you arrive at clinical studies yet, or
not yet.
in the process?
We are close, but not yet. We are very close.
I think this is the next step. Having a patent, then you are close to go to clinical studies or not?
Can you repeat the question? I'm sorry. Maybe my internet connection, because there's an
No, it wasn't your connection. It is the setup of the room. We have multiple microphones and speakers.
I'm sorry.
So there's an echo on your side then. So the question was, what was the question? So when you have a patent, then my colleague thinks that you must be very close to a real solution.
Yeah. Well, actually, yeah. Yes or no? Because we designed another compound, and that is way effective than the previous one. So we focus on the second one right now. So we believe that the compound hopefully.
Don't you want to sell the first one first and then sell the second one later?
Well, yeah, because the biggest problem with these inhibitors is actually the toxic hepatotoxic effect, and the second one is really low. So that's why we are really interested on that one. So maybe the first one we may not even use it. So the second one is effective. So maybe I can send you the structure and you can find me new targets, and because the biggest problem we have is in here. We don't know the effect of metabolites because I don't have access to LC-MS, and I really want to know the effect of metabolites because I know the effect of gene expression, proteins, but I'm missing metabolites, and I'm more interested the effect of metabolites, maybe you know, in COVID studies, I find out that like these glucose receptors are the expression of glucose receptors are decreased, but the symport glucose receptors are functional, and that decreases the sodium concentration. So that's why I'm really interested in working in metabolites, because as I said in my talk, like the triple negative breast cancer displays both Warburg effect and oxidative phosphorylation. So if I know the effect of metabolites, you know, I can design new compounds. So maybe you can help me in that sense, because I don't have much information about metabolite, and I know that there are at least 100,000 of them.
So I'm sure your friend or our common friend Talat Yalcin can do the LCMSMS. That shouldn't be a problem. The other question I would like to ask is, who's doing the molecular docking for you?
Actually, a group in Turkey, they are helping me. His name is Yusuf Sert. I'm not an expert, although I have Yasara software, but I mean, so I'm asking his help, but well, that's limited.
Yeah, because I'm wondering, a lot has been done in the last couple of years, so that those tools and the 3D prediction of protein has so much improved that that might fuel your further research. I don't know. So I'm
Yeah.
not an expert on that either. So that is
Well, that will be quite help, because his major is not molecular biology. So sometimes, you know, it's hard to understand your colleague because you're not in the same study field. Yeah, that's
definitely.
maybe I can find another collaborator that he or she can understand, you know, my research. So that will be nicer.
Great. Yeah. So thank you very much, Yusuf.
Yeah, I appreciate. And
I will contact you via email later.
yeah, thank you.
Some other questions. So we have to progress here. So thanks again. And it was very informative. And I hope we will have you again perhaps next year. We'll see.
Yeah, hopefully. We'll see. Yeah, thanks.
Thank you, Yusuf. So indeed, the next speaker on the list is myself. Du kannst von da reden, aber dann hören alle anderen das auch. So we will not turn on the other microphone in the room again. It makes too much Hall effect. Yeah, so I will be talking about also cancer. Interestingly, I'm not an expert on cancer, obviously, but and I will come to that during my talk, we did a little study, and let's see if I can. Yes, so we see the complete screen, and all we did is look at RNA-seq data, and we realized that perhaps it's not enough what we do at the moment, and we might have to consider other targets than just say human. And that is something I would like to get into now. So how did this come about? Well, I'm professor for medical informatics and bioinformatics at the Hochschule Ruhr-West in Mülheim an der Ruhr in Germany, and teaching in the study program Health and Medical Technologies. That's a bachelor course, bachelor program, and it's 6 to 7 semesters. Typically, students need 8 semesters, but the program is for 7 semesters. So, and there are many reasons why it takes 8 semesters, and even some are in the, I don't know, 15th or 16th semester. But yeah, the average takes 8, and that's because of the course load and because of the diversity of information we provide. So one course that I teach is medical omics, and that's an elective course. And in that course, students have to provide portfolios. Portfolios means every week or every significant amount of new information, they summarize that information, make an image or make a something and put all of that together into one portfolio. So at the end of the course, which is a block course, that's a half a semester only, but twice as much per week, they're done. So there's no further examination or anything, it's just a portfolio, and then it's finished. There's one alternative path to this portfolio, and that's a computational data analysis. So whether they do a portfolio or they write a research paper with a computational data analysis, that is equal for me. So last semester, one group chose the alternative path and provided a computational data analysis. The 2 students were Alpay Khan Erbay and Taha Ilga. And these 2 had an idea of looking at viruses and looking at RNA-seq data analysis. Before we come to the details of what they actually did, we have a look at the course Medical Omics. Due to the background of the students, I cannot assume that they have a very large amount of knowledge about molecular biology, cell biology, or anything of biology. And therefore, I start with the cell. Yeah, there are different types of cells, we all know that. So we go into that area first, and then we go from the inside out, from genomics to transcriptomics and proteomics. And then we see the components, right? We have the genome that encodes all of the possible components. We have the transcriptome, which actually then either provides components or encodes for further components on the protein level. So with these components, we can then see how they work together. That is the gene regulation part. And that kind of concludes the first part of this course. And then we come to markers. And I don't think I'm turned on, am I? Wait just a second. Or, oh, come on. For the people that are not here, they might not see any gestures or anything. So perhaps I should turn that on. Okay, great. So we came to the part where we have the components together. Oh, this is very low response of the computer, sorry. Yeah, now we're there. Okay, so we have the components, the genes, the transcripts, the non-coding RNAs, the RNAs that are coding and then turned into proteins. And all of these form together one big network that performs cell homeostasis. So with that network in place, we can then look at whether there are problems, diseases. Often, when a disease is manifests itself, some of the components are either not there or in low amounts or something bad happens, and these can be used as markers. So we go into markers, and then if we have markers, if we can precisely tell what is going on in a particular cell, then we come towards precision medicine, and we can then see perhaps we find some way to treat a particular problem very easily in one patient because they produce a certain marker, and not so easily in another patient where they don't have this particular marker. So this is the overall structure of the course, from the cell to precision medicine. Obviously, we have to stay on a very high level. We cannot go into detail everywhere. But when we talk about genomics, for instance, we talk about both things, the genome how it works on the molecular, in the cell, on the molecular level, and how we measure it. Yeah? Next generation sequencing and other probes that we could use, plotting, etc. So those things are discussed. Oh, yeah, there I had a slide for that. Yes, we have DNA sequencing. We could also do think about microarrays, RNA sequencing, mass spectrometry, and each of these things by themselves is a very complex story. So I filter and provide the current information, and we skip all the problems that happened in the last, say, 70 years of mass spectrometry, and just focus on the cherries on top. Then we go into other things that can be done to actually test whether something works, like silencing, to see whether the gene that we predicted to be doing something actually does it when we take it out of the equation. And then we look at the end at pathways, so how things are regulated, how things are put together. And these are the, in part, experimental methods and in part computational methods. We do not talk about computational methods much in the course. That's only for the groups that actually choose the computational path. The others are kind of shielded from that part. So back to the course and the course work and the examine that they are going through. So weekly assignments, they have to bundle into portfolio. I encourage them to provide these weekly assignments in the week that they produce them so that they can get feedback from them. And some students do, many students don't. And then they provide the portfolio at the end and are surprised sometimes why it is not so good. But the ones that actually make use and provide it in the week and then improve it are following the feedback. They are usually not surprised. So the other part that is more important for the talk today is the computation analysis. And they only have a subset of the weekly assignments. They do have to do the cell. They do have to do the regulation and the markers so that they get a general understanding of what is going on. Plus do their computation analysis, but they have to see where it fits into the overall program. Their submission is a little bit different. It should be like a manuscript that we could select, submit to a particular journal. And they cannot measure anything, so they have to rely on existing data. So I encourage data parasitism, and but I think that's a good thing to do. At least we can duplicate the findings of people that did it before, or in this case, we find something completely different. So what was the motivation of this group? I mentioned the names before, Alpay, Khan Erbay, and Ilgataha. And they were intrigued with viruses and also interested in cancer. So that comes together in 1982 already, where some team, or Harald Zuhausen's team, already detected papillomaviruses and established a connection between cancer, some particular cancers, and papillomaviruses. In 2008, he was awarded the Nobel Prize for Medicine for this, and this is one of the motivations why this study was done. So after that 1982 first measurement, there have been about 10 other human papillomaviruses associated with various cancers. These viruses are non-enveloped double-stranded DNA viruses, and they belong to one class, the Papillomaviridae, and there are many papillomaviruses underneath that particular group in this class. So many things are possible that we might not know yet about, but we were interested, or the students were interested in looking at these two things: cancer, non-cervical cancer in this case, because that's where the papillomaviruses were first associated with, and then
any papillomavirus, not just the HPV-16. Another issue is that these viruses are very easily transmitted, not as easily as many other viruses, but mere skin contact is enough. And therefore, this virus may be spread through the population. Another issue is some are completely without symptoms forever, and others actually develop symptoms. So as long as you don't have symptoms, but can still spread the virus, you're a threat. We all know this routine now after years of COVID. And this is just another such virus that you may transmit without even knowing that you're infected. So the question that the students wanted to address is whether these viruses can be detected in randomly selected RNA-seq samples of tumors from the Sequence Read Archive SRA at NCBI. And one more, there was some information why or how they choose their data sets. It's not supposed to be cervical cancer, because that means that there's a very high chance that it is papillomavirus. So obviously, they had to do some data analysis, and I encouraged them to have negative and positive controls. So they put, they selected data where they were pretty sure that it would contain papillomavirus, or where they were pretty sure that it would not contain papillomavirus. And these are the controls that they selected: some pneumococci measurements, some anthrax in cell culture. Luckily in cell culture, not somewhere else. Why you would select this data set, I don't know. But yeah, positive data were, for instance, viral circular RNAs in cancer. And there was some annotation that there would be human papillomavirus in there. So they were pretty sure it would be there. And then a direct analysis of HPV-16 that must be definitely a positive example. For the other sample selection process, they had small laptops, so they could not select huge amount of files, and they could not select huge amount of data, obviously. So the data was restricted to be between 5 and 10 gigabytes, and it should not be cervical cancer, and then it should be different cancers. So if they would use, they didn't want to use 5 times, 6 times lung cancer, but they wanted to use different things like lung cancer, leukemia, and so forth. So RNA-seq data analysis is it's a huge topic. And some years ago, Conesa et al. Offered a way of how it should be done. And this is also approximately how it was done. There was no trimming and clipping, but at least there was the quality control. But we will see from the quality that there was not really necessary to trim and clip. And since Bauteil 2 was used, it has an internal clipping. So that was also not really necessary. Therefore, we shortened this pipeline a little bit. But other than that, this was followed. And what we see here is we need RNA-seq data as input, and we need reference data. So the reference data is one of the things we will talk about. But which tool are we going to use? How can I ask students in a course that is half a semester, 8 weeks, no, not even 7 weeks long, to do quality control when there are 20 tools that perform quality control? Are they going to check every tool? That might take longer than the course. Yeah. How about clipping? There are another 20 tools, so perhaps we cannot finish the first part of the pipeline within the course. So yes, I can tell them which tool to use, but that would not be a very scientific process, right? That is quite the opposite. So this is clearly a problem that we face. And it goes on. Read mapping. There are more than 50 short read sequence alignment tools. Have a look at the Wikipedia page that I put here. There are at least 50 of them, probably like 80 more. And no one knows which one to use. We could say, let's use the one that is most cited. but then we don't know if it's a positive or a negative citation, right? So that's one thing. The other thing is the older the tool, the more citation it acquires. So that this is really not a measure that helps us. And if we then go to quantification, there are also many tools such like HR, but some statistics are really outdated. So still using maybe reads per million or so. These things probably not up to date anymore. So how should we choose the components? Let's avoid this discussion at this point, and we will just say which ones we use. That's FastQC, Bowtie2, and SEM tools, or not we, the students used. And this is something for a paper that I just submitted and that is accepted. So you will see in a few weeks, months maybe, how to discuss this further with the tool selection, etc. So this is the workflow, genome assembly, and RNA-seq data, quality check, alignment, and then we look at the counts and the coverage. We see here the quality for the negative and positive data. There's really nothing to complain about. They are perfect, and I don't see anything that could be wrong with them. We have in the selected samples, 2 samples that have a little bit of quality issues. The third one in the top and the second one in the bottom, but not really worth mentioning, and the few 100,000 reads that we may lose in the hundreds of millions of reads, I don't think they are worth pursuing with extra clipping and quality filtering and so forth. So if they don't map, they don't map, and that's okay in this study. If we want to reproduce this and extend the samples, then perhaps we would take a more distant, more precise approach. But here in this case, that is totally acceptable. So then we come to the first analysis, and that's the controls. And applying the workflow to the controls reveals that we find some papillomavirus in all of them. Yeah? However, let's have a look at that in more detail. Do not focus too much on the counts, because the counts are relative. They are specific to one particular sample. They are specific to the measurement. They're specific to the machine. So we cannot really compare the counts or the reads per million or anything that results from the counts among samples. So what I would like you to focus on is the highest coverage. So what the highest coverage means is how much of the virus genome was covered with reads. So 100% means that every nucleotide of the virus was also found in the sample. Yeah? Which then would mean that the virus is actually in the sample and probably viable and probably reproducing itself and so forth. So their absolute counts are also high, totally. And that is also important, but as I said, not comparable. I think that the coverage, however, is comparable. And if we look at the positive and the negative data, then we see that the negative ones are, in this case, 10% coverage. And that is also a small human papillomavirus fragment. And the other one is less than 1%, which can just be some spurious mapping. So we will not find 0%, that's for sure, if you have a larger virus database. But we will find a very small percentage, and 10 and 1% is something I would say that is not an indication that the virus is in the sample. But looking at the other two, the positive controls, we see that we have 99% and 76% coverage. And there I would be alarmed and say, okay, that could really mean that, yes, the virus is in the sample. And why would it be that we only get 76% coverage, although we have a huge number of counts compared to the other one? Well, the problem might be that that particular virus is not directly in our sequence database, and we're missing a part of it. And again, where does the sequence database come from? It's not easy to find a virus non-redundant sequence database. So what the students did is they compiled a database themselves. They went to NCBI, Googled for searched NCBI for the genomes, the fully assembled ones, and then used that to create a database. Then they went to the samples, and there they had 6 samples, and you can look at the left part, the study, and then you can have some idea of what the studies were about. And some of them, for instance, used cell lines, and in these cell lines we find 100% almost contamination, let's say, with papillomavirus. But I later checked the literature, and yes, that seems to be expectable. So basically, after some time, these HeLa cell lines become contaminated or were contaminated from the beginning. So others have found this as well. And in 4 of these 6 samples, we find some of the papillomaviruses that they had in their database. So that is, in my opinion, a very striking result. And again, we judge that by the coverage. And the coverage in every case is above 90%, which means that most likely the complete virus is expressed. What we do find sometimes is that there are some parts missing. Here is a very small example where one particular, this could be a nucleotide or so, is missing. But we do have, maybe two, 3 slides later, we do have also an example where a big fragment is missing, and still that would explain a lot. So coverage again means how the reads, and you see that in the lower part of the picture very well. Each of these gray or blue or red bars is a read, and how they align with the selected genome. And you see that the genome is more or less completely covered by reads. So if we look at the most frequently detected HPV genomes, those are the ones, and as I said, the students produced the genomic database themselves. So what they didn't know is that a lot of these things are very, very similar, and some of them are fragments of another. So what we do see is that NC001357 is actually the full-length HPV18, and that the other 2 are actual fragments of the HPV18. So those are partially completely covered and sometimes not covered at all, those fragments. And a bigger part of the NC001357 is missing, but it's always the same one missing. So it may not have anything to do with that there is no human papillomavirus in there, but that the one virus that is in there we did not have in the database completely. So this is the part that is missing, and would be very strange if there wouldn't be some virus that this would completely map to. So clearly, for future performance, we would have to create a better non-redundant sequence database that covers perhaps not only human papillomavirus, but other viruses as well. And with that, we could then have students perform such type of analysis, again, maybe with a different point of view, and make them successful in such a very interesting, intriguing kind of work. This is their poster. Thank you very much. Do you have any questions?
I have a question. Which course was the bachelor? Okay.
It's a bachelor course.
But
And
which area? Or what is
medical
the-
omics is the,
Oh,
is the
it's
yeah.
Bachelor of Medical Omics.
No,
Oh,
the
no.
no, the bachelor is on health technology, more or less, but that's a crossover of engineering, computer science, and yeah.
So you have students from all of these backgrounds.
No, you don't have students from any of those backgrounds. You have students without background that come and take the bachelor, and then some of them succeed and some of them go other directions. So yeah, we're very tight on time, so perhaps we should progress. The next speaker is Wissam Gültekin, who is a PhD candidate at Bielefeld University. Wissam, are you there?
Chat.
But I can't. Why can't I? Yeah, now you can speak, Wissam. Thank you.
Hi, can you hear me?
Yes.
Okay. Hi, everyone. You should be able to see the slides, right?
We do. Thank you.
Okay. So today I'll try to talk about complications in statistical analysis of RNA-seq data. And I didn't know that Jens would cover some of my talk, but that's okay, I guess. So before going forward, let me just mention that the transcriptome is the interpreted functional elements of our genome, right? So when we say transcriptome, it's we mean the complete set of transcripts, including messenger RNAs, encoding RNAs, small RNAs, and so on, in a cell. And we are pretty much we are interested in their quantity because their quantity pretty much affects this specific developmental stage of a cell or physiological condition. So the widely used technology to analyze RNA, the transcriptome, which continuously changes in a cell, was microarrays. But with the advent of NGS technologies, next generation sequencing technologies, RNA-seq became the choice. And the most common aim of RNA-seq analysis is the profiling of gene expression. And how we do that is we identify genes and or molecular pathways that are differentially expressed between two or more biological conditions. And how we do that is mainly we might interfere with a gene in some select samples, let's say with RNA interference. So in the image, it's pretty much summarized the whole procedure. We select some cells and we try to extract the total RNA, and we further fragment these RNAs, and then we ligate those and convert these to complementary DNAs, and we feed them to sequencers. And at the end, we get millions, maybe billions of reads that will be feeding to our analysis. So before going on, I want to show you two links in specifically two links. One is there are lots of methods. Illumina is maybe the let's say more mostly used technology in RNA-seq studies, And when you come to this page, you can see that they show you lots of technologies that you can choose for your specific study, for your specific interest. And there is this poster, which is, of course, very extensive, but again, it is very interesting to have a look at least. And it shows lots of protocols for RNA-seq analysis. So let me go back. So generally, what we do with RNA-seq data is fits into this workflow. But as Jens just said, there are lots of differences. So what we have, once we feed our data to these NGS technologies, we have our data, which are called reads. Each fragment become a read. In some cases, we would like to we would like to have the fragment read from opposite directions. And if that is the case, then what we have at hand would be paired end library. So after what we have in hand, we have to perform some quality controls. And as Jens just said, most of the time, in recent years, most of the time, it's just a quick look at what you have at hand, because the accuracy of our NGS technologies became more and more significant and reliable, and the efficacy is rising. But all in all, let's say 95% of the time, you have to perform a quality control step. And then what you do is you map your data to a reference genome or reference transcriptome. As I said, this is not the only way. There are some applications, some methods and analysis that you can perform without a reference genome or a transcriptome, a de novo mapping, for example. But generally, you map your data, and then what you end up once you map your data to a reference genome or transcriptome is you have a what is specifically said count data. Counts data means is it's a extensive and an exhaustive table that show you which reads were mapped to which gene and their quantity. I will show you an example later on. And once you have these count tables, then you perform your differential expression analysis. And of course, you can go on. You can go on with this analysis, but this is the generalized version. So as I just showed you, the RNA-seq technology might output single end or paired end reads. What single and paired end read means is, if you have single end reads, that means is your fragment was read sequenced from only one direction. And if that is the case, you have a read shown in here in blue rectangle. And once you try to reference this to your genome, that might result with some ambiguities, because the fragments might be longer than the read itself. What I mean is a fragment that you feed to the NGS might be, let's say, 300 bases, but at one go, sequencer mainly reads like 50, 60, 70 reads. So your fragment might be bigger than the one read that you have. And also your reference genome or transcriptome might have lots of repeats. And in that case, you won't know how you can put your alignment in correct position. But if you have paired ends, that means your fragment is read from both directions. So you know that there is a known area within the reads. So once you have that information, in the same example, you would say that, okay, first repeat is more compatible with the first repeat in my genome, and that way you would be much more confident in the assigning read to chromosomal location on your reference. So quality control step begins with the raw data itself, and what we have once we have our reads from NGS is FASTQ files. FASTQ file is the modified or improved version of FASTA files, and as you know, FASTA files have the identifiers and then the sequence itself. Here we have two other lines, and we have the separated line and the quality values. And these quality values are existent for all the sequences, and they are encoded in ASCII, which means it's you encode a numerical value with 1 byte, which is, as I show here, it can vary from exclamation mark to many uppercase or lowercase characters. And since we have lots of NGS technologies, we have lots of different combination of these matrices. But the most common, mostly used matrix for this kind of for this kind of situation would be FRED, which was introduced in like 90s, but it's a solid matrix for this step. And as you just can see, we have lots of differences within FRED as well. Illumina is the more prominent and mostly chosen technology in RNA-seq analysis, and itself it has at least 3 different applications. So this is one of the problems with our computational biological problems. I didn't make sense there, but you understood, I believe. So once we perform this analysis, what we want is our quality data, our sequence would have at least Q30 quality. With Q30, you would see this Q30 everywhere in lots of papers. What Q30 means is the base call in the sequencer. The base call was can be 1 in 1,000 calls that could be 1 errors, but other 999 were pretty much confident. So we can believe in this sequencing base call with 99.9 accuracy. So what we mostly choose is Q30 in our base call controls. Other than that, we have to remove our adapters because we introduced these before feeding our data to the sequencers, and these tag sequences might be the artifacts like the multiplex identifiers. They might be adapters, they might be primer sequences. So we have to remove them. And as Jens said, in recent years, sequencers, while spitting out the data, remove this at one go. But again, we have to look at those. And we have to remove short sequences, and we have to remove sequences. What I mean by sequence is the read, of course. We have to remove reads with lots of M bases, which means that sequencer was not confident in calling any of the 4 bases we are interested. So once we have our data, we have checked the quality, and we perform some steps to ensure the quality of our data. what we have is a set of reads, and we have a reference genome or transcriptome, and we have to perform a mapping. And in this simple figure, we see that what we would like to see is our reads somehow overlap with our reference genome, and these overlaps would provide us with lots of more information. But as I just said, we would have repeated regions, highly variable regions in our reference data, and also, as I just told you, we might have sequencing errors, or we might have shorter reads than other reads in our hand. So our reads might align to multiple regions at our reference genome or transcriptome. So what choice do we have? And basically, we might have 4 options. We would say, okay, map the one read that wherever it maps, which is not, of course, a right choice. We might say, okay, map to whichever region randomly. Again, this is not a feasible solution. We would say, okay, let's discard the read, and sometimes we do that. But the better or best choice would be we would have to have some scoring for each map, for each read, and highest score would be our choice. So we need scores. As I just showed you, one read might map to multiple locations in our reference data. And to overcome this, we have to have some scoring schemes at hand. And I would like to show you, to summarize the whole idea, I would like to show you a nice representation coming from University Illinois. I didn't know if I pronounce it correctly, but anyway, so we have a sequencing sequence alignment game, and the idea is you see the reference on top and the read at hand at the bottom, and these are the same scores that were used most of the time, including BLAST itself. And then what you do is you shift bases to introduce gaps, and optimally you would like to achieve this score. Right now, before doing that, it was 0, and it is 0 again. So I moved 1 and I still have 0. If I move this, as you can see, I introduced 2 gaps, and these are the scores, but I achieved 60. And if I have this kind of placement of my read, then I would get higher score, and I would say, okay, if my read is aligned to this position, that is the correct one. So let me allow me to make more sense in showing you this. So this is a real data that I fed to 4 independent mapping tool. My reference is at the top, this one, and my read is this. So these are all results coming from real tools that you can find online. And just by look at it, there are significant differences, but they are all the real outcomes from each tool. So as Jens said, which tool we can use? There are, as Jens said, there are lots of tools, but the idea should be we would the first step would be we need to be let's say we must know our data. We must know our data, the technology itself, the NGS itself, and the read count, the divergence, the dispersions of the reads, and so on. And we have to investigate. We should not just pick a tool with the citation count and so on, but we have to read. There are lots of comparison studies across, we have to be aware what we choose and which tool to use. And sometimes the hardware we have little bit pushes us to some tool because there are lots of computational expenses when you look at the tools themselves. But again, there are lots of tools, and we have to be aware that each tool, Yusuf Malik is requesting to annotate. Okay, I approve. Yusuf Malik wanted to annotate. Was that a mistake? I don't know. Okay.
Probably a mistake. We some continue.
Okay. So, what I can advise would be, if we're talking about RNA-seq and you would like to perform your analysis, mapping analysis to a genome, you should pick a tool that is spliced-aware mapper. It's because, as we all know, this is a biological phenomenon. Once you have your analysis coming from mapper, what you can do is, what you should do is feed your data to some other tool just for looking at the data and analyze it. So this is again a genome browser, and these genome browsers show the data in different tracks. So at the top you can see the chromosome itself, its sequence, base sequence, and at the bottom you see the mapping results. Each horizontal line shows a read, and I highlighted one of the reads here, and it shows from which point to what point, and the strand itself, and so on. By the way, when I mentioned strand here, so again, another recommendation would be, if you would like to increase your efficacy or the accuracy of your analysis, I would recommend using stranded RNA-seq data. And I might comment on this later on if you like, but from because of the time restrictions, I should go on. So once you have your mapping results, again, you should look into the data, and this is one of the ways of looking at the data. Here we see that most of our results, our reads, were mapped to CDSs, and this is the scenario that we would like to see. But of course, this is not whole indicator that our data was nice, our mapping strategy or analysis workflow was okay. But again, we should be able to, we need to have an understanding what our analysis churns the data, what is our current situation. Yeah, so here again, I show you another point that we should be able to understand while analyzing our counts data. Here we see that, as we all know, genes might consist of lots of exons, and our reads might be aligned to one gene, but again, it might be on the left side of the figure. You see that one read was mapped to the gene correctly, okay, fully okay, but it was mapped to different exons. So in this case, we would not count 5 reads matched to gene 1. Rather, we say, okay, we have 5 portions, but 2 portions are coming from the same read, so we count this as 1. At the right side, you see a little bit, let's say, complicated or advanced version. Again, you do not
have to be very quick now. You're running out of time.
Really? Okay, I am sorry. Okay.
ran out of time actually.
Really? Oh, my time says 20 minutes right now, but okay, sorry. So what you end up is samples on the columns and genes at the rows. But here, this is the row results coming from your mapping and read counting analysis. These are not meant to be compared directly because these are raw data. And I'm speeding up a little bit. And I would like to show you just a simple example here. We have 2 samples and we have 4 genes. And for the sake of the simplicity, I'm not talking about their length and so on. But here you can see that we have 42 reads for each sample. But because we might be comparing one tissue with another one, or we might be comparing the same tissue, but in one sample we down-regulated one gene, or we silenced it completely. The composition, we also alter the composition of other genes as well. So here, if we do not account for this biological condition, reality, we would say, okay, all the genes were differentially expressed. But reality is not like that. We have to account for the reality that once we feed the sequencer, we have a set time period. So for sample 1 and sample 2, let's say we have 1 hour for each run. So if we do not have GMD in sample 2, the sequencer would be we would read more. We would have more time to read gene A. This is called sequencing depth, and if we normalize for that, as you can see, the only differential expressed gene would be D. So because I have to speed up, there have been lots of efforts to overcome this problem, and there are lots of studies that specifically say in differential expression analysis of RNA-seq data, the greatest impact is normalization part, because there are lots of things that you have to account for. In this scenario, we have to account for sequence depth itself. At sample 1, we have only 22 reads mapped to our reference. Sample 3, there are 291. But that doesn't mean the genes were differentially expressed. So there have been these first announced or first published statistics, but these are pretty much they are not being used anymore, or they are being used to a shorter or limited cases, let me say. So if we use RPK here on the left, we do not bring sequence step to equal proportions for each samples. And in this case, it is harder to see which gene is differentially expressed between the samples. Here on the right, we see TPM. By the way, I didn't write M because it is million, but with our data, I used scaling factor of 10. That's why you can see sequence depth 10. And now we can easily compare samples within with one another. And here we can see that, okay, GMB was present 0.6 times in sample 1, but 1.5 in sample 3, and we can come to other conclusions. And as I showed you just before, library composition with the advent of the NGS technology, became more and more prominent, and we have to account for that. And there are lots of tools and workflows that is made for this specific problem. There are lots of tools, and some have some assumptions that the counts data would have to fit in some distribution, like normal Poisson and so on. But okay. But again, in recent years, more cited and more recommended versions of this are listed, like here in this slide, Agile, DSEC, and so on. And we have non-parametric approaches which do not base their analysis on the distribution, and we have some other modeling approaches. In fact, I would like to speak more, but I am running out of time. So shortly for
Wissam, thank you. We have to stop at this point, otherwise we lose the complete sequence. There would be many questions from my side and from, I see people taking notes from others here as well. So, but we will have to save them for later.
Okay, I am sorry.
That's okay. We have our next speaker on the list, Lena Raupach. Maybe I have to turn her microphone on. Yeah, we should be able to hear you now. You should be able to speak now.
Yes, do you hear me?
Yes, we hear you. Thank you.
Then I will share my screen.
Wissam, you may have to unshare your screen for Lena to be able to share her screen. Yeah. Now you can try.
Okay. Yeah. Hi, everyone. To introduce myself, my name
Don't
is
see the screen yet.
No?
No.
I will try again. Do you see
Yes.
it
Now we see it.
Okay.
Thank you.
So, yeah, my name is Lena Raupach, and wait. And I'm also a PhD student of Bielefeld University, and I am working at the company ID Berlin in order to implement my dissertation project. The project I will talk about is called GenVada, and it is a conception and implementation of a pharmacogenetic dosage database. That was a bachelor thesis project of my student Hauke Noah Goers, which he finished this year in May. And I will continue this project within my dissertation, which is about using such a pharmacogenetic database in an existing medication check in hospitals. First, I will talk about the motivation and the database concept, and after that, in the method part, about filtering academic papers for manual data extraction, and then I will give a brief example and use cases, and finally an outlook. When it comes to prescribing medication, hospitals are moving more and more in the direction of precision medicine. Therefore, medication checks are already being used in many hospitals in Germany and around the world. A special form of precision medicine, which is not yet routinely used as standard in drug prescriptions, is a pharmacogenetic medication check. This involves providing a warning for drugs if they are metabolized differently than usual due to a particular gene variant in the patient. For example, as you see in the right figure, desipramine and imipramine are metabolized by CYP2D6, And if the patient has a gene variant of this enzyme causes a slower metabolism, the patient could get an overdose of the drug if the dose is not lowered. A pharmacogenetic check would decrease the risk of inappropriate prescriptions caused by different drug metabolism and thus benefit the drug therapy. At the core of such a PGX check is a suitable database that contains the necessary information. Since the state of knowledge is constantly changing due to ever ongoing research, a database is needed that is constantly growing and collects every piece of information from these scientific publications. And this is a small weak point in PharmGKB's dose guidelines. Some of them are outdated because they are only added to the database after a complex revision process. Medication checks should also score their results to quantify the significance of the dose guideline. The data from the G standard from the Netherlands, for example, does not provide such a score. The medication check should show understandable results of recommendations for their prescription and how safe it is to follow that recommendation. And this is what GenVeda is designed to achieve, a complete knowledge base that grows with each new paper published and that provides a score that can be used as a guide. The database concept contains the entities gene variant, level of evidence, drug, GenVeda, reference and source, and the GenVeda entity connects them all together, and the reference entity will contain the information from the papers, for example, dosage recommendation, quality level, or patient specifics. And every reference has a source with a PubMed ID. Now to the method part, filtering academic papers for manual data extraction. In the first step, the challenge is to collect the drug variant pairs from special dosage requirements and to create the database. In the second step, suitable papers with specific dosage recommendations are found and made available. The general aim of the program is to find all papers whose information should be manually entered to the database. The filling of the database should happen in following projects. Okay, step 1. PharmGKB offers various kinds of pharmacogenetic data, for example, guideline, drug label, or clinical annotations. In the figure below, you see examples of the clinical annotations. For GenVeda, we need the drug variant pairs, so the variants and the drug. With their pharmacogenetic associations and their level of evidence that describes how proven this association is. In addition to that, the annotations have a phenotype category there that describes the category in which way the variant affects the drug, for example, dosage, efficacy, metabolism, or toxicity. And in the first step of the algorithm interacts with PharmGKB and looks for drug variant pairs with a level of evidence 1 or 2 and a phenotype category of dosage or efficacy. And the resulting drug variant pairs are cached to a text file along with the annotation ID, the PubMed ID count, and evidence count for step 2 of the algorithm. So here. And before the second step, the pairs and the other existing information from PharmGKB is stored using Spring Data for database access. So the database exists after step 1 for the user to be filled with information from papers. the papers that are likely to contain information about the dose adjustments are filtered out of pub data in step 2. The second step begins with using the drug variant pairs of phase 1. The text file with the variant drug combinations is read in and put into objects. For every variant drug object, an HTTP request to the API of PubData is called. And PubData is a web-based system that not only offers biomedical PubMed abstracts and PubMed Central full articles, but also annotates their content into biomedical concepts like species or a specific drug. It offers access to 3 million full-text papers and 30 million abstracts, and various of them are of interest for pharmacogenetic dosage recommendations. And to find them and to integrate them into one database is the goal of GenVeda. Yeah, you can either use the API or download the data via an FTP server from PubData. And in the prototype of GenVeda, the API is used to search for the papers. And yeah, the PubData server is able to export papers in JSON format. And the number of posts to the API is restricted to 3 requests per second in order to not overload the server. That makes the program relatively slow, but I think it's okay to sacrifice the velocity as the program is about creating the database and it can run overnight or in the background. The output JSON contains the papers and some information about them, like the species. Here, another filtering step is done by sorting out papers which are not about Homo sapiens. This is one of the bioconcepts that are marked by PubData. Some samples of the API requests show that the PubData search sometimes return papers that have nothing to do with the inserted search terms, the variant drug, and the key dose. So in the JSON file, the text search algorithm needs to be filtered out papers that do not contain the terms of interest. So if a publication has all the filter criteria, it is possibly useful for the pharmacogenetic database. So the PubMed ID and a link to the source of the paper is stored in an output text file. And from then, they can be accessed and the information needs to be extracted manually. Unfortunately, the dose indication of the drugs in the papers are not unified. Sometimes they are in grams and sometimes they are in milliliters. That is difficult to normalize in the database and to draw a conclusion at the later analysis. But one important information is, does the dose need to be increased or decreased? And this is easier to normalize and can be found in many papers. And finally, an own quality level is calculated for each database entry to see how valuable this information is for the pharmacogenetic check. And the quality level is calculated from the following summands. First, the number of paper found for the drug variant pair by the GenVeda algorithm. This is highly weighted with a factor 0.5. Second is the publication date. If this is empty, the default value will be 1990, because due to technical progress, newer study might have more accurate results. And this is light-weighted with a factor of 0.005. Third, the number of PubMed IDs that supports this clinical annotation, or the PubMed ID count from PharmGKB, with a factor of 0.02. And fourth, the difference between the evidence count and the PubMed ID count from PharmGKB with a factor of 0.3, where the evidence count is the number of annotations supporting this clinical annotation, including variant annotations, guideline annotations, and drug label annotations. And yeah, this is an example of the GenVada database. In the columns drug name and gene variant here, those values are entered automatically from with the algorithm in step 1. And the columns dosage, title, and ethnic group or patient specifics are entered manually after extracting the pharmacogenetic information manually from the papers. Yeah, now I will talk about use cases. My research topic is to develop a PGX check, an extension of a medication check by a genetic component. In case there are annotations from a patient's drug variant combination, a warning should be displayed. And my goal is to show the doctor exactly the issue of what drug variant pair, of that drug variant pair, and an understandable and reliable recommendation. The quality level is intended to allow the doctor to assess exactly how necessary a dose adjustment is and how devastating ignoring the warning could be. The inclusion of a PGX check in the prescription process will meet with a lot of resistance and skepticism, I think, because it is new. Therefore, a valid database that you can rely on and that provides all the necessary and important information for a dose recommendation is essential. And this database would have use cases in the industry as well as the pharmacogenetic research, because it integrates a large set of knowledge from scientific papers, and it is machine readable and could be used by medication checks. Yeah, to the outlook, the prototype has shown that there are already enough papers for such a database, and from this point on, it still needs to be filled. This is a lot of manual work. An option could be to use a natural language processing algorithm to run over the found papers and to fetch all the information automatically. But since medical data is involved, an NLP algorithm might be too error prone. Yeah, this topic is discussable. The first question when it comes to creating a database is who will maintain it. Keeping a database up to date is a lot of work, and though it is highly valuable for the pharmacogenetic check in the medication check. So the answer is that the database could become a part of the medication check. In the case of the ID Pharma check of ID Medics, it could be maintained by ID Berlin. Also, rules for a dose guideline check could be derived from GenVada data. This derived dose guideline rules can then, after manual validation by pharmacists or doctors, for example, used as knowledge base in a pharmacogenetic medication check, for example, ID Medics. Also, further software tools could generate statistics or analysis of the database, as it is a collection of information from papers. The database will continue to grow, and of course, conflicting information will be included. Any scientific knowledge in the field of pharmacogenetic dose recommendation can be valuable. They may be inconsistencies, or they will be inconsistencies and contradiction when it comes to collection of all scientific findings in pharmacogenetics. but a collection of them would make this knowledge useful in many ways, I think. And that's it. Yeah, thank you for your attention.
Yeah, thank you very much. Questions? But I have one question. Who is entering the manual information at the moment?
This was a bachelor thesis project. So when the prototype was developed, it was my student Hauke. And yeah, so probably in the future it will be me. Yeah.
And ID Berlin has a interest in having such a database and continuing to support such a database. Is that right?
Yes, they have an interest in the medication check or the pharmacogenetic medication check. And yes, and thus the database as knowledge base. And the validation of the data could also be done by colleagues from mine. They are doctors and pharmacists, and with the help of my colleagues, I could create a usable database.
We do have a prominent text miner in the audience at the moment who will speak later on today. Perhaps something more could be done with text mining than you already did to further streamline the process.
Yes, this would be very interesting. Yeah.
Okay, Malik Youssef is the name, so perhaps you can get in contact with him later.
Mhm.
All right, yeah, thank you very much. And we continue with another talk, and this is done by Dilan Kiran. Dilan, are you available, ready? I will make you able to speak. Yes, now you can speak.
Hi, can you guys hear me?
Yes, we hear you, we see you, and we see the slides. Perfect.
Okay.
And now we don't see the slides anymore because
And
they're
then,
on the other
yeah,
screen.
we see the speech now.
Yeah. Shift, Windows button, and left or right arrow pushes the file to the other screen.
It's not a problem. I will share without the writings. And you can see now, I think.
Yes.
Yeah, perfect.
Okay. Hello, everyone. My name is Dilan, and today we will present to you our research outcomes of the project named Criteria for Evaluation of Workflow Management Systems for Scientific Data Analysis. My colleagues are John from Izmir Institute of Technology and Dr. Jens Elmer from Hochschule Ruhr-West. The presentation will consist of 5 parts. We will start with our motivation, then continue with a literature review, feature selection, and criteria application, and will end by sharing our conclusions. The human body has trillions of cells which each contain thousands of different proteins. The proteome term consists of all the proteins within a cell. Throughout human life, the DNA remains the same. However, the proteome constantly changes. This exemplified by the distinction between a young crawling caterpillar and a grown-up flying swallowtail butterfly, as you can see on the slide. The protein network is crucial for cell homeostasis, thereby is also important to determine the health of the organism. Hence, the study of proteins, which is called proteomics, is a very important part of medical omics. Measuring proteins expression by RNA-seq, as Jens Elmer and Vita mentioned before, or establishing their identity by mass spectrometry is allowing us for a deeper understanding of proteins and their networks. Although the steps involved in proteomics are highly simplified, they clearly lead to a complex endeavor. The same thing also applies to a subsequent data analysis or any other scientific data analysis. The data analysis workflow shown here only presents a part of possibilities that can be analyzed using proteomics data. As you can see from figure here, both genomics and proteomics analysis involve many steps to interpret the data into meaningful information. The data obtained from an experiment or from a database must be converted according to needs of the tool that will be used. This step alone may create problems as changing tools or workflow management systems mid-analysis may not be possible. Therefore, the initial choice of workflow management systems may become crucial for the whole analysis. To see which workflow management systems are being used in the field of informatics, we did a literature search for a number of citations through PubMed. The table here is an abstract data, and our analysis is not limited to these workflow management systems. As you can see, KNIME is leading with 2,289 citations. Although the general usefulness of workflow management systems could be considered proportional to number of citations, but the citation numbers may not be a good criterion to choose workflow management systems. There are also other factors affecting the number of citations, such as commercial status of workflow management system. An open source and public workflow management system is expected to get more citations than a commercial and closed source one. The age of system and associated publications also contribute to number of citations. Here you can see our methodology. We start by our research by seeking workflow management system from Google and PubMed and listing them according to their citation numbers on PubMed. Then we did a literature survey on features of workflow management systems and try to identify most mentioned features in the articles. Subsequently, we build a consensus table to list the features according to their mentions in the chosen 8 articles. To choose our criteria, we did not base them only on their consensus, but also base them on our definition of the features. Lastly, we tried to find the level of application of each criterion in KNIME, GenePattern, Galaxy, Pegasus, UniPro UGene, SnakeMake, Nextflow, and CLC Bio by reviewing their official sources.
Well, in order to comply with the presentation time, we limited our consensus table to the features that we later deemed critical or important, but you can see the full table once we publish our paper. We calculated the consensus scores by dividing the number of articles that mentions said criteria to the total number of articles we reviewed. So the scale is from 0 to 1, and these are the features we later deemed critical: reproducibility, reusability, fairness, versioning, and security. Although security had a consensus score of 0, we chose the feature as critical simply because of the importance of privacy of data, especially in the health sector. This may the security may create unpleasant consequences if the patient's data gets leaked or accessed by an unauthorized person. And these features are later deemed as important criteria: graphical user interface with a consensus score of 1, flexibility with 0.75, scalability with 0.75, shareability with 0.63, and computational transparency with 0.50. In order of decreasing consensus for each column, we offer the following features for discussion as viable criteria for the evaluation of workflow management systems. For simplicity, we will focus only on critical criteria and explain only critical criteria in the following slides. First, the reproducibility. Reproducibility defines the ability to generate the same exact result with the same data set using the same workflow with identical parameter settings, regardless of other environmental choices, such as the operating system. And also, reproducibility must be ensured throughout all the components of the workflow and not just the overall workflow, because in the worst case, the components made up the workflow may generate random results, and the result that must be identical and not just as close as could be in a biological experiment. Second is the reusability, which defines to reuse the same workflow for intended purpose or a related one. The first step of reusability basically covers reuse of the workflow with the same intention with a different data. The second step covers shareability, not shareability, sorry, reusability, sharing the same workflow with other users of workflow management system. And this could be achieved by platforms that provides access to the workflows via digital object identifiers, DOIs. The third step is the platform independent sharing of the workflows, but this seems elusive due to the large number of platforms and previous attempts at platform independency, such as Shiba. Another question is how narrow the scope of the workflow is and whether it can be used for a related purpose. For example, can a workflow used for alignments of RNA-seq data from an Illumina measurement accept different types of RNA-seq data measured with other machines from other vendors?
The third criteria is FAIR. FAIR is an initiative to make scientific data findable, accessible, interoperable, and reusable. The findable and accessible parts of FAIR are often enforced by journals during the publication of manuscripts that include data by requiring its deposition in publicly accessible repositories. For data, interoperability can be achieved by using standard representation of data, such as MZML in proteomics. The reuse of data typically depends on the level of documentation and metadata that was collected. The better data collection and data are annotated, the higher is reusability for other purposes. From workflows perspective, we mentioned reusability before. We tied findable and accessibility in with shareability, which is important criteria that unfortunately we can mention. And we covered interoperability as part of reusability. Versioning is the ability to track modifications made to a workflow within a workflow management system. Both change and editor should be trackable, similar to versioning systems such as JIT. Stable versions need to be identifiable so that fairness is ensured. Our definition of versioning has three aspects. The first aspect is versions of workflows. With current systems, versioning extends further to the underlying tools whose versions may not be always tracked. The second one is about versions of workflow management systems itself. Workflow management systems also undergo changes, and it must be clear how to access stable versions to combine the right tools with the fitting workflow and workflow management system versions. Lastly, versions of underlying tools used in a workflow are included. Often, workflow management systems provides access to command line tools produced by third-party tools. Swapping out one version of such third-party tools against another may lead to significant changes in the data analysis results. Hence, all of these aspects of versioning needs to be tracked.
And lastly, security, which defines the ability to protect access to the dataset, workflow, and the outcome of the workflow. That should only be accessible to certain verified individuals. As we mentioned before, the privacy of the data, especially in the medical omics, must be secured because it can generate unpleasant consequences. Also, the companies may want secure access to their work because they may want to develop a commercial product, so they need to restrict the access until the release of the product or only to the paying customers. And as same with the reproducibility, security must be ensured throughout all the components of the workflow, and not just the overall workflow. All components, for example, third-party tools must be secure, and there should not be any backdoors. For simplicity, we will focus on 3 workflow management systems, Naim, Galaxy, and SnakeMake in regards to critical criteria application. Keep in mind that we didn't actually use these workflow management systems, but only reviewed the literature. So some of these workflow management systems might be underrepresented or overrepresented in the table. However, the main takeaway message is, although there are many workflow management systems available to use in the internet, none of them apply all the criteria we have chosen, important or critical. To conclude, there are more than 100 workflow management systems available to use, and there's no way to compare them because each have their own different features, each have their own advantages and disadvantages. There is no list of criteria to assess these features. We performed a literature review, extracted the features, defined the criteria, applied the criteria to the most popular workflow management systems in bioinformatics. Certainly, there's a large room for improvements, and hence we are open to feedbacks. Well, that is all for now. Thank you so much for listening to us. For those who are interested in this review, you can read further information once we publish our paper. If there is any questions, we will be very glad to answer them.
Yeah, thank you very much. They can always contact us as well instead of waiting for the paper. That's always good opportunity. Yes, so since we have no, we don't have an overlap. No, you're perfectly in time for the next speaker, Malek Youssef. So I would like to thank you two again. That was a very nice presentation. And the paper will come in now in 2-3 months, perhaps. We will see more about it. Yeah. All right, with this, I would like to invite a long-term collaborator of mine, Malek Youssef, who is working in multiple fields, including text mining and the application of machine learning to different areas in biomedical research. So this is something we will hear about today. And Malek, if you're ready, I may have to turn you on, right?
Yeah, I hear you. Thank you very much. Do you hear me?
Yes, thank you very much. We hear you. We see your slides. Everything's perfect. So please.
Okay, I just try to make myself with the annotation. Okay. First of all, thank you for inviting me for this interesting workshop. I would talk about part of our research, actually. Most of our research was maybe in microRNA, the sequence and stuff like that. So I would like to introduce the concept or the approach. We name it GSM, Grouping Scoring Modeling. The title, actually, we said that it's about integration of biological domain knowledge to machine learning based gene expression data analysis for selecting effective genes. Actually, maybe I will introduce you, if I have time, not just in the gene expression. Also, we have applied this approach in text mining, and we published the manuscript. We're still developing stuff in this direction. So let me see. Maybe I should just turn this annotation. Okay. How it's supposed to go next? Cannot go next. So that's what I should do. I should eliminate this one. Okay, now good. Okay, so biological integrative model. If you are interested in more details, I would recommend to you to read the review paper that was written by Borjo, Apishka, and me. It's about actual application of biological domain knowledge based feature selection on gene expression data. We survey actually different approaches about integrating knowledge. So we actually use the term biological knowledge because we are working in biological data, but actually what I'm introducing to you could be in general integrating any knowledge in a given data set. So most of the algorithm actually try to look to part of the picture. We try to develop the tools that actually somehow consider the big view, like the image that you see here. So mainly we work in gene expression data that it's known to be extremely high dimension data. So for simplicity, the data could be consist from rows that represent the sample, where it could be cancer, healthy. So there is always the column class that represents the label class of the data, could be negative, positive, and the columns actually host the genes. So the merit of our approach is actually came by that most of the existing feature selection, or we will introduce not really feature selection, we will use group selection, maybe this is the right phrase, where actually we're borrowed from the field of computer science, statistics, and they apply it in biological data. But in our approach, actually, we try to consider the biological domain or the knowledge about the data in order to improve or maybe come up with a new outcome. So we call it to consider biological domain knowledge or pre-existing biological knowledge about the data. For example, if the data is actually gene expression, about the gene expression consists from genes, so one of the things that we can use is, for example, microRNA. So the microRNA targets genes. This is actually the tool that the first tool we published in this category, Jens and me. So the traditional approach is consists from doing some pre-processing, applying some feature selection, resampling, classification. There is a lot of algorithm around. People continue to suggest different kinds of machine learning algorithms, supervised, unsupervised. There is a lot of pipeline that can that you can follow in order to perform some statistical or machine learning analysis. And then the outcome will be somehow a list of significant genes. That then actually the traditional approach is to take this list of significant genes and go to map it, do some enrichment analysis, somehow map it to, for example, public data set based like MIRTAR base, KIG, disease, SNPs, and then find the connection, do some biological interpretation, and so on and so on. So if you look here, you see there is two steps or components. They are somehow separated. The first one is doing the data processing and the machine learning or statistical approach. The second one is to use the knowledge, the biological knowledge that's represented by database. So when I say biological knowledge, I mean somehow this knowledge is represented by some database like MIRTAR base, KIG pathway, disease association genes with the disease, SNPs, and so methylation, and so on. So the second advantage, so one of these papers that actually we start to see that people talking about taking advantage of multiple published gene expression data, combine this together, aggregating this knowledge, multiple data sets, actually will increase the statistical power in a defined small subset of genes. So that in the end, we try to find a small subset of genes in order to maybe serve as a biomarker. So the integrative approach actually is something that is rising now, and we are shifting from what we call it from what's called data-centric to incorporating additional biological knowledge. So we are actually not using just the numbers from the matrix that is given as a matrix. So the data set is actually represented, the data represents as a matrix of number, as you saw, as I showed you before. And then actually we start to add more biological knowledge in this term. So traditional approach actually in machine learning, there is given to some, for example, two classes, patient, control. So the linear discriminant analysis is actually to find the linear classifier that separates these two classes. And in simple way, actually to find the line, actually it's not line, we're not talking in two dimension. In multi-dimension, we're talking about maybe a hypersphere or something like that, or a space that actually can separate the two given classes. So in simple way, actually we find this equation where is the G to Gn is the features, the genes, and the weight actually is the weight that we discover by solving some mathematical equation. And if you look deeply to this equation, you can just figure out that the weight actually somehow have information about the importance of the genes. So genes that actually associated with low weight, they are not important for the model. For example, just consider that genes associated with the weight with 0, the value 0, then we can say, okay, this is not important to this equation. So somehow the weight here actually gives some information about the importance of the genes. So one of the approaches suggested by Isabella, she's a student of Babnik, is to perform what we call recursive feature elimination, where they actually apply fit the model by support vector machine, and then they redo the process by actually by removing the weak features or the weak genes according to the weight, and then actually redo that, fit the model, and rebuild it again until you end up with a couple of genes or a couple of number of features that you are interested in. Okay, so if you just start to look to the literature, you start to see there is something what they call it clusters-based feature selection. So this is the phrase, clustering-based feature selection. In our terminology, we use maybe a group-based feature selection, because clustering actually is based in some similarity in terms of the values of the genes, but in our approach, we actually consider the pre-existing biological knowledge in order to put the genes into clusters. Maybe the clusters is not the correct word to use, so we call it groups, so set of groups. So our biological integrative approach, we call it GSM. Let's just try to explain it somehow in general. So we call it integrating knowledge. So assume that you have something like that. Assume that you have a matrix that consists from genes in the column and sample in the rows, and you have this is magic column that actually contains the information about the class. So the question, and also there is some another knowledge that is given by maybe what we call it actually knowledge represented as a table. So the problem is that we have gene expression, or again, it should not be just gene expression. It could be any text mining data, another kind of data set that you have in general. And there is a knowledge. The knowledge is represented as actually a matrix where there is a group, and the group actually somehow have information about the genes. So I love to make some annotation. So if you look here, you will see that this group actually have some knowledge or association between the genes that somehow they are grouped in the group number 1. And if you look, all of the genes actually somehow related to this matrix. And another group actually consists from different set of genes, group 4, and so on and so on. Just to make it simple, we have the same number of genes. In reality, this is not the case that the group have the same number of genes. Okay, just for visualization. So the things then how these two knowledge, the gene expression or the data, and the knowledge that represented as a groups. So how we can use it in terms of machine learning. Let me see if I can clear my annotation. Okay, I become now. I start to how I can. Okay, now good. Okay, so if you look to just some simulation, then actually the group, there is association between the knowledge and the data set. This is the group number 1, group number 2, group number 3, 4, and so on, so on. As I said, the size of the group, the size, I mean the number of genes that belong to the group should not be the same. So how we integrate this knowledge? So the integration is happening in this way. Actually, for each group, actually we extract sub-data set from the original data set. So now we represent the knowledge given by the group. We extract a sub-data set from the original data set, and then actually the next question is how we can give a weight or score those groups. So now we are scoring not individual gene as happening by the traditional approach. We're scoring or we're giving a weight or ranking the group. So the idea is to, first of all, to represent the group by a sub-data set extracted from the original data set. And then the next question, actually, now you have in your hand a sub-data set. Because I mean, we have a sub-data set, which is actually including the classes, because we extract from the original data set. So we have also information about the class. So what we have now in hand, what we call it actually two data sets, a two-class data set. So if you have a two-class data set, you have a knowledge about the class, and then you have a knowledge about the sample and the genes, then you can use any machine learning or any statistical approach to give a weight for those sub-data sets. So this is actually what we trying to suggest here. Let me continue. So the question then how we can score each group, and there is actually a lot of way to do that, maybe using statistical, machine learning, and so on, so on, so on. Okay. So one can suggest also something like that, use some maybe a statistical approach by using the t-statistics maybe, and another approach is using machine learning. This is actually what we are using, the machine learning. There is a new study that we might study to see the influence of the way of the approach that we use in order to group and to do some scoring. So in general, what we have something like that. We have a two-class data set. We split the data into train and test. We keep the test data part not involved in any stage before building the classifier. And then actually we use the knowledge from the train part and the knowledge given by any public database like Mir-Tarbase, KIG, disease, SNPs, methylation, or even the user can give his own knowledge about the data. We extract for each group a sub-data set that represents this group. Then we move all of these sub-data sets to a scoring model. The scoring model performs some scoring in order to give weight importance for each group. We rank it. Then we use the top ranked group in order to train the model, and then we test it by using the test part that was splitted and extracted from the original data set. We report about something new, the performance table, which is actually maybe somehow it's a traditional approach to perform the performance, but there is something unique that we perform the significant groups, where actually now we report about significant groups rather than significant genes. And the group actually represents some biological knowledge. And we report about actually the group name and the genes that actually associated with this group. So this is something new that we report in our tools that we doing. Okay. So in general, this is what we are doing. We create sub-data set, then we move it to somehow machine learning algorithm in order to score it. And just keep in mind that we score each sub-data set individually and not in the same time. Even we now also try another approach to do that by considering different combination of a group. So each sub-data set that created become input to the scoring component. One of the approach that we try to do is to split the data again, the sub-data set, into some maybe sub-sampling approach, random, k-fold, whatever. And then we take part for training, part for testing. We train the model random forest, or could be suppose vector machine, could be any algorithm that you would like to test. We repeat this. We call it inner cross-validation. We repeat it a couple of times, collect all the performance matrix, and then we somehow report, we give the score, the accuracy score that was collected, or the average You also can use precision, F1 recall, and then we give this as a score for the sub-data set, which is actually, this is the weight that we calculate each sub-data set. So now we report, we collected all the scores. Now actually we consider all of these groups, we rank it, we use the top groups in order to train the model. We start a project that we did not finish. Maybe we need to find the next student to work on that, to find the association between the size of the group and the score. We start this project with Jens and Borjo. Her student actually, she's busy with other stuff. We would like to continue this because this is very important question that we need also to see, and or how we can eliminate the effect of the group size in the score. So in the end, actually, we report, as I said before, we try by using the top groups, we train the model, we test, and then we collect. So we try using the top first group, then the second, cumulatively, the third, and so on, until we reach some specific threshold that we determine in the beginning. So if you just look to the visualization, you see that we start by the top group, then which we consider the top 2 cumulatively, then the top 3 groups, and so on. And we try to report about this kind of table for the user. So actually, GDNet discover multi-disease. One of the projects that actually we submitted, and it's still under review, is how we can use, for example, gene expression and the diseases. The outcome is something very interesting. So some example of pre-existing biological knowledge, for example, we use the MIR-TAR base. So the outcome is we report about the top groups. So the group, for example, microRNA that target genes, now we give this information that was not provided by other tools, and then we ask for a given gene expression data set that studies specific disease such as lung cancer using grouping, scoring, modeling, will show the role of microRNA. Even the microRNA is not part of the data set. So using the knowledge by the biological knowledge can provide additional information that actually was not designed by the experiment, because the experiment is actually a gene expression data that we have in our hand. For similarity also, we report about the disease association between the genes and the disease. Then actually we report the role of the disease with other disease that we have in our data set. Drug gene interaction. This is a new project with Professor Fee from UNC that actually we start to see some good results in this direction. So there is three parts. Sorry, there is three categories how to consider integration of biological knowledge. The first one is to use the fully data driven from the data, like we published the SVM-RC, SVM-RC-ER. Now we are working with RC internal, RC scoring centers. Actually, we get the knowledge that given by, for example, that genes that are correlated, they having similar function. So in order to do that, we use k-means in order to group. So here we are not really using external knowledge, we're using biological knowledge and using the values that given by this matrix. So this is one of the ways that we use. And as I said, also as a report, we actually were involved in this competition of CAMDA, ISMB. We developed actually text net topics where we use the topics as groups in text data mining. So this can be very useful way also to deal with text mining. We published this study and we're still working this project. The other kind of integration is using pre-existing biological knowledge. The pre-existing biological knowledge is given by some database like KIC pathway, gene ontology, MIR-TAR base, disease, and so on. For this kind of knowledge, we actually developed SVM-RNE, MATI with Jens, Cognet, GDNet, still under consideration, gene ontology, PR path, also Jens involved in this project, also is also under consideration. MERNA, DISNet, microbiome net, and so on and so on. So again, the knowledge represents as a table of groups, that each group actually consists from genes. All of this actually, you are invited to look to see this kind of application. The third one actually is using multi-omics. Now the input for our model is more than one omics data set or more than one gene expression. For example, we have developed a tool called the treatment, where we consider 3 omics data sets. The 3 omics data sets were microRNA expression, messenger RNA expression, and methylation expression. The question was how we can integrate this 3 omics data set to create groups and assign it, and then grouping, scoring, and modeling. All of these tools actually also you can have it from the GitHub. Here we start also to add more information to let the user use it. All of these tools actually written in 9. Again, thanks to Jens that introduced it for me. I never knew what does mean 9, but the time that Jens introduced it to me, we shift to 9. 9 become the tool, the programming language that we are considering and using these days. So the first, how many time I still have?
You still have time, no problem. 15
Okay.
minutes, I guess, yeah.
Okay. So the first, in the beginning, actually, the first algorithm was developed in Wistar Institute, where I did my postdoc there. So I think that when I was working with gene expression analysis, applying all of this traditional approach, then I saw that, for example, Luis and the other postdocs, they take the significant genes and they somehow send it to some tools in order to find the knowledge about these tools. And during the discussion, I learned that genes that function similarly, correlated, they function similarly. Then I come up with the idea why we are not clustering the genes. So actually, all of the ideas start that by we have data, we cluster the genes. This is actually the groups by using k-means. And then the second approach is to do some scoring for this cluster groups, eliminating the least significant genes, which is actually somehow related to RFE. Then we go back, we cluster again, scoring, eliminating, and so on and so on. This study was published in BNC Bioinformatics. The algorithm called recursive cluster eliminating for classification and feature selection. Recently, actually, now we start to dig deep and suggest other tools, other algorithms that I will talk just recently. I will talk later on. So the output of MRC look like that. For example, we report about the most significant cluster. This is actually the accuracy, sensitivity, specificity. We calculate all of this information, and then accumulatively the performance about the 2 TABS clusters together, the TAB 3 clusters, 4 until we end until 90. And also we report about the significant genes that show up from this tool. We compare it for other tools so that MRC providing good and similar results as before, but the good thing that actually the genes that we discover is somehow different from the traditional approach because we keep clusters. And if you look here to this heat map, you see there is some clusters in our heat map that you cannot see in the traditional approach like RFE or PDA or another linear algorithm. Recently in 2010, actually we start to see the influence of the ranking approach, and we see that using actually the rank or the scoring component can actually influence the performance. Somehow increases dramatically. We also did this study and we published it in F1000 research. Okay, recently actually, as I said, we started a project, Jihan, a PhD student in Abdullah Gul University, Borjo from also Abdullah Gul University, with Professor Bahjat Kakish from UNC, to see if we actually introduce a new approach called internal elimination. So we go actually deeply inside each cluster and eliminate the least genes in each cluster. We saw that actually this approach can also somehow enhance the performance and provide least number of genes in the end of the process. And as a future work, we wrote in SVM-RCE that the success of the SVM-RCE method in classification suggests that gene interaction network or other biological relevant matrix that group genes, those based on function parameter, might also be useful. Actually, this is something that we predict, but we didn't have so much time until we, in 2019, Jens and me, we start a project of MATI, where we integrate the microRNA knowledge, and we suggest this tool. We publish it in bioinformatics. But just to let you know that we are not we're having fun between 2009 and 19. We were working actually in other field of microRNA genes, target sequence. We did a lot of work. Most of this work actually was done with Jens and his students. Okay, so as mRNA, actually also we use the knowledge about network. This project was done with my previous PhD supervisor, Larry Menevich from Haifa, and Luis. We continue that. And also we suggest this approach where we group the genes using some GXNA that actually, the GXNA actually put the genes into networks. The networks actually represent a group, do that. And we publish this again. So this is actually the results of SVM-RNA. I will not really now start to explain all the results. You can just read it from I will just make a quick survey about the different tools. So MATI actually was something very interesting. This is the first project that we use pre-existing biological knowledge. All of the tools before actually we use somehow knowledge from the data. Now actually we add knowledge as a database from microRNA. So we have gene expression, we have microRNA groups, machine learning, and the microRNA expression. Actually, this is something that we wrote that in somehow how to use also microRNA expression for gene expression and microRNA expression. This is another tool that we published. We call it MIR-CorrectNet, where we integrate the two OMICS data set, gene expression and microRNA expression. This is something that also was done. So the idea for MATI is that we use the knowledge given by MIR-TAR-base, where actually we have a knowledge about the gene microRNA that target genes. So this is actually the knowledge given as pre-existing biological knowledge. We use it in order to group the genes, and then we apply the whole process GSM. If you look here, you see that the knowledge is MIR-TAR-base. We create sub-data sets that each sub-data set represents a group from this database. And then we move it to scoring, modeling, and report about the performance and output significant group. The interesting thing, actually, this is where we start to plot also the group size to see the group size, because this is very important to see if we have a huge number of groups and the size of the group. The interesting thing in this tool that now you start to see something additional to the traditional approach rather than SVM-RCE. We start to see significant groups, but microRNA expression is not part of this data set. So this is the unique of our approach GSM, that now we report about the role of microRNA in this specific data set, and how that we try to predict the role of microRNA in this gene expression without even having the expression of the microRNA in this specific data set. So traditional approach search for significant genes for the model classification. MATI search for significant genes for the model classification that are associated with microRNA should be targets of at least 1 micron. This is the difference between the traditional and MATI. MATI search for these groups that actually somehow related to this knowledge that we use it as microRNA target in this tool. So the next tool was actually how we can now suggest a tool that uses the microRNA expression and the gene expression. So this is actually the first study was that we use 2 omics data sets. So in order to do that, the first question was how we can generate the groups. So we use the microRNA and the gene expression in order to find the groups by applying some maybe correlation matrix, Pearson correlation. So the genes, the microRNA that have, for each microRNA, we calculate the highly correlated genes, and these genes actually somehow represent the group that associated with this microRNA. So the group now actually is extracted from combining the 2 dataset microRNA and gene, and in this way actually we extract the groups, and then we apply our approach GSM. So the main idea here, the question was how to create the groups using the 2 omics dataset. And again, if you look to the mere correctness, we have output of significant genes. We have also the performance table and the most significant microRNA. Now we have 2 outputs, the significant genes, the significant microRNA, and we have the performance table. That actually the biology now is very important to make sense about all of this information. So for that, actually, we have a collaboration with Mitra and Christian from Jefferson. They actually took our output and they make sense about the result. They found very interesting biological results. For example, MIR21 5P was actually ranked highly in our analysis, and it seemed to be also very important in cancer. Again, I would suggest you to read the manuscript about that. So as I said, now the third part, the third tool that we're presenting now is how to combine 3 omics data sets. So this is actually one of the things that actually we just recently created a tool, and we done with it. We wrote the manuscript. This project is also with Borjo from Abdullah Gul University and Steve Merriam from UNC. During my visit to University of North Carolina, we established this kind of collaboration. So the idea is very simple, maybe not, is to use the 3 omics dataset in order to create the groups. And then when we have the groups, then we can apply the GSM approach. So the idea was in this way that we use the first 2 omics dataset, the microRNA and the CPG or the methylation, in order to find the pair, the highly pair correlating between microRNA and CPG. And for each pair, actually now we start to build the group. So the group actually was built from the 3rd dataset. All of the genes that are highly correlated with microRNA and this is the CPG from each pair is actually attached to this group. So now in this way we create the groups. Now we have the groups, we apply it in the gene expression data. So if you just notice, we use the 2 omics data set to create the pair, and then we use this pair with the 3rd omics data set. But in the end, we apply the GSM, the SM actually in the gene expression data. So this is the target data that we use it for the for the SM component. Again, we split the data into train, test, create the groups, fit the model, random forest, or could be any other machine learning algorithm, and we report about the significant groups, performance table, and significant groups here. So this is the figure of treatment that actually show all the combination about the 2 omics data set, and then we integrate the third one, create the group, scoring, modeling, and report the output. This is the figure that we use. Okay. Okay. So this is actually the output of experiment. Again, it's similar to what we have before. The top group, number of genes, average genes, accuracy, specificity, sensitivity, area under curve, and this is actually the second, the cumulative 2 groups, the cumulative 3 group, and so on until we reach 10 group. Actually, we decide 10, but anyone can continue with these numbers. We, according to our experience, that we did not see any improvement after even 5 or 6. So maybe the best thing is to we will try to find the most predictive number of groups in order to report it to the community. Okay, I will not go deeply in the analysis of this tool. I will go over another tool. One of the tools that actually, as I said, we start is to use the GSM in drug-gene interaction. This project also with Faye from UNC. Golsom is a PhD student. We almost create the group. There is a lot of good stuff here and unique stuff about the drug. So the drug actually that target some genes, as we have seen here, they are actually the groups, and the genes that are associated with this drug, they are actually the content of the group. And we start to use this for our GSM model, but we face some problems. There is a lot of common information between the drug, and we have facing some supergroup, subgroup, stuff like that. We're still dealing to solve all of this problem, but we start to see some good results in terms of drug interaction with genes. Pathfinder was another tool that was with the actually.
You may have to come to a conclusion now.
Okay, so my conclusion is I just want to represent that we use some pre-path also was the microbiome.net, also another tool that we use in microbiome.net. So what I try to say that this approach, the GSM, I just will get to the end, can be applicable just not in gene expression data, could be also applied in different kind of data, for example, text-net topics. And I am sure that this approach can be applied in different medical data that you have. So we are open for collaboration. If you have some ideas how to integrate or specific data that you would like to apply this GSM, we will be happy to do this kind of analysis with you. Thank you very much.
Thank you very much. Are there any questions? A lot of tools, yes. There are a lot of tools, Malik, that I think all the tools are running, yes. So now, I personally don't have questions at this point anymore. It's a bit late for me, but thank you for the comprehensive talk. Yeah, so all we have to do now, Ralf, is thank all of the speakers.
Thank you very much.
And wir hören dich nicht. Moment, just a second, Ralf. We don't hear. Geht das Mikro?
Okay. Also, thanks to all speakers. And what I said is, I hope that we will have a workshop next year live in Heilbronn, and live will be much more better than this Zoom conference. So a lot of talks, and I think Zoom is not so easy to understand. I like more to see people here in front and then we can discuss. It's not so nice, but thanks again. And perhaps you have questions what we are planning. So one plan is next year workshop, and perhaps we will found this work group as soon as possible. We will see. That's the key plan.
Yeah, so we also have the IABC that for some reason didn't happen this year. So that was unfortunate, and we do have a medical omics workshop in the IABC as well.
Yeah.
So hopefully that will both happen again next year, and
Yeah.
both should be in presence.
Yeah.
Right.
Yes, and then we can ask GMD to have this official work group. And
I think it's about time that we formalize the situation.
Exactly,
Yeah.
exactly. So this was a plan years ago, but Corona produced a delay. And having live workshops, I think then we will have more participants. That's the plan.
We did have quite some participants changing over the different talks today. There were like 50 live views, different people apparently, and there were like up to 20 people in the online and combined here audience. So I think that's a pretty good draw for such a small medical omics workshop.
Yeah,
right?
I could not see this number.
Yeah.
So this is very positive because also if you have a live workshop during the GMDS conference, you will not have more than 20 participants. So that's positive. And perhaps a few participants have questions. You can also send questions via email to us or ask now.
If you have any questions, you can ask now.
Yeah.
But yeah, Ralph and I are typically responsive to emails within a few weeks.
Oh no.
Just kidding.
Yeah. Perhaps someone is interested in cooperation to build up this work.
Yeah, okay. So with this, and we would like to close the session today.
Yeah.
And we hope to see you again soon in another medical omics workshop or some other meeting.
Yeah.
Thank you very much for your participation and see you soon.
Goodbye, press the button.